Does anyone here like baseball? A couple people. We have this cool thing in baseball called the 7th inning stretch. When you've been sitting there for a long time, everyone gets up and does a little dance. You guys have been sitting there about an hour and a half, and I think we have over an hour to go. Why don't you all stand up, give a little stretch, get some circulation in your legs, and then give us a minute, and then maybe we can focus back on some talks. All right, doesn't that feel good? Are you all baseball fans now? So I'm assuming from the title that I was given that I was meant to talk about arrhythmias. So that is what I will be talking about with a few extra things sprinkled in. But the title that I sometimes use in other places is Genetic Discovery in a Million People, Where Do We Go From Here? So I want to just make sure that I appropriately acknowledge all the people who really do this work. Somebody makes a joke that graduate students do their research by coding, say in Python or something. Postdocs do their research at Excel, and PIs do their research in PowerPoint. So these are the people who are hands-on doing the analysis every day, and they're an amazing team. I absolutely love working with them. I'm not sure if Wei is here. Is she here? She was going to come. Anyway, so first, why do we study the genetics of human diseases and traits? My goal is that through our work, people will be able to live healthy, active, long lives. And the place where I think our team can make the most difference is in avoiding premature death due to heart disease, as you just heard about from Peter Donnelly. This is a picture of my grandfather, who was actually born in Barnstable in Devon, who is just turned 95. And this is another picture of my brother-in-law representing a healthy, active individual. So how can we do this? We can hopefully identify genes involved in a variety of different traits, and we identify these genes through naturally occurring variation in humans. We take advantage of nature, and these genetic variants in genes may become leads for either prevention or treatment. And as you just heard, very nice description, we can hopefully also predict who would most benefit from preventive lifestyle changes, medical screening or treatments such as statins in individuals who are at highest risk of adverse events. So this is a little cartoon of how I see the sort of scientific pipeline. We start with enrollment of individuals in something like a biobank, of which UK Biobank is obviously one of the world leaders. There's this pipeline. There's the DNA technology that you've heard a little bit about. There's the quality control and analysis pipeline. We always, in talks, gloss over the quality control, but this is, the actual most important and sometimes most challenging part of ensuring that you get good results out the end. I use a little tap to indicate statistical rigor and deciding on which things that come out of your results at the end you deem to be true results. Then there's a bunch of variants and loci that sort of fall into this pool, and there's actually a bit of art in the science of picking out which of these genes you might want to follow up in very expensive animal experiments, often done in mice, rabbits, and occasionally pigs. Hopefully these lead to new therapeutics in clinical trials, which will then help treat disease. We're now getting better at hopefully also identifying who will most benefit from these therapeutics. So I'm sure you're all very aware of this, particularly before starting the UK Biobank, but biobanks amortize the cost of genetic studies across many traits. So the study that I've been working primarily with is the Hunt study from Nord-Trøndelag in Norway, and we are users of the UK Biobank very happily. So typically a biobank will have genotypes and phenotypes. In Hunt, we have about 1,400 traits we've been analyzing in a mere 70,000 people. We had the slight advantage of having low-pass genomes from 2,200 individuals, so we could call indels and use these ancestry-specific haplotype reference panel combined with other publicly available reference panels to impute variants into the 70,000 individuals from Hunt for Norway. We analyzed 1,400 diseases and quantitative traits that came from hospital ICD codes as well as blood measurements as well as some survey data. One of my graduate students came back from a weekend and we said, what'd you do this weekend? And she said, oh, I analyzed 1,400 traits in the cloud, which was much more impressive than anything I did on a weekend in graduate school. Also cost $60,000 of cloud computing. Of course, when you're trying to understand the results from 1,400 traits, it's a little bit complicated, so we built a results viewer. Do I have 5 minutes left? Do I go to 11:45? 5-ish? Okay. I'll go quickly. I think we started 3 minutes late, so just saying. So Wei is a super talented graduate student working with a super talented biostat faculty at Michigan, Sean Lee, and together they realized that the state-of-the-art software, when UK Biobank first came out, couldn't really handle a biobank of that size. So they developed a new method that not only incorporates logistic mixed model, previously a linear mixed model had been applied to dichotomous traits, which isn't quite correct. They used a saddle point approximation to deal with the statistical issue of having an unbalanced case control design, which is typical in biobanks. So back in the old days when we collected 1,000 diabetics and 1,000 controls, you had equal numbers. The statistics were a little bit more robust. But from a biobank, you might have 10,000 type 2 diabetes cases and then a whole bunch of controls, which leads to some statistical difficulties, particularly when analyzing rare variants. And then they used some optimization strategies that had previously been applied to this type of software. So for example, the state-of-the-art software at the time UK Biobank came out was Bolt LMM, which is a linear mixed model. And there's a lot of false positive results, particularly for very unbalanced case control numbers. And SAGE cleans up all those false positives. So SAGE is available. Wei also analyzed on a different weekend 1,400 phenotypes from the UK Biobank and made the results for these 1,400 phenotypes publicly available. So I'm supposed to talk about atrial fibrillation in my last few minutes. Atrial fibrillation is an arrhythmia. It's a very common arrhythmia that affects many people, particularly in the older ages. Around 2010, it was primarily thought to be a defect in ion channels. So we, together with collaborators, including Decode and UK Biobank, and published data from the leaders in the field, including Patrick Eleanor, we collected together 60,000 atrial fibrillation cases and 970,000 controls, and we got a little Twitter love for having the first GWAS to reach 1 million samples in discovery. We identified 163 independent risk variants at 111 loci, and through a variety of bioinformatics methods, prioritized 163 putatively likely functional candidate genes. This is what the GWAS plot looks like. It looks an awful lot like Manhattan, which is nice. And this is what those functional candidate genes look like when aggregated by biological process or biological pathway. And what was really exciting was seeing a number of, really expanding the biology beyond just ion channels. For example, a number of genes that cause congenital heart defects also appear to be impacting age-related atrial fibrillation, as well as a number of transcription factors involved in cardiac development. So it's clearly much more complex than simply an ion channel disorder. When we performed a FEWAS, so we took each individual in UK Biobank, developed their polygenic risk score for all atrial fibrillation variants, and in fact for all variants across the genome, and performed a FEWAS, we saw association with a variety of phenotypes, many of which are cardiovascular phenotypes, including atrial fibrillation, cardiac dysrhythmias, et cetera, sort of validating the result. But the MD PhD in my lab, Jonas Nielsen, who performed this study, said, you know, when people come into clinic with atrial fibrillation, they're seeing a cardiologist, they get an EKG, they're gonna have lots of related ICD codes related to that diagnosis. So when we excluded the 7,000 people with atrial fibrillation, the FIWAS looked extremely different. So that's something to keep in mind when you're doing FIWAS. You might want to exclude the cases because of this ICD relatedness that happens. Surprisingly, we found genes related to abnormal heart development. We found genes that were highly expressed in fetal heart tissue, which again expands our understanding of the biology of this phenotype. We went on to look at a specific locus, the myosin heavy chain locus, where there's both a fetal and an adult form of myosin heavy chain 6 and 7. And we examined these genes in a fetal rabbit heart model. So by inducing heart failure and the heart failure model actually developed atrial fibrillation. So after, oh, there's a pointer. After burst pacing, the normal heart developed normal sinus rhythm, whereas the heart failure model could not. And in the heart failure model, we see increased expression of the fetal form in the left atrium. And this is in the damaged heart, the heart failure model that develops atrial fibrillation. We see heterogeneity of the MYH7 and MYH6 genes. And those genes impact the contractile velocity. They have different rates. So one's a fast model, one's a slow. And when you have heterogeneity of which myosin heavy chain is expressed in different cells, you can imagine this could lead to atrial fibrillation. Whereas it's much more homogeneous expression in the right atrium or in the control heart. So similar to what Peter just showed, we see a different lifetime risk of atrial fibrillation based on the polygenic risk score. So just to sort of come back around to the main point, biobanks allow us to study many phenotypes. So within the Hunt study, we identified a rare loss of function indel that causes a 42% increased risk of fracture, bone fracture, and a 32% risk for having a bone mineral density below the clinical threshold for being medicated. We looked at estimated glomerular filtration rate as a marker of chronic kidney disease. This was prior to UK Biobank releasing related phenotypes. We found 53 new loci. We've been examining thyroid stimulating hormone, which identified 66 new loci. We looked at liver-related blood traits, including liver enzymes measured in 50,000 people. Identified 89 coding variants, of which 11 are loss of function in a specific gene. And 17 of these have an impact, a massive impact on risk, greater than 1 standard deviation. And then lastly, atrial fibrillation I just talked about. Another project I don't have time to go into, but my group is part of the leading group of the Global Lipids Genetics Consortium. We've collected information from 1.2 million individuals from about 250 cohorts globally. And now that we can add in UK Biobank, now that the lipids have indeed been released, thank you. It was 2 months away for about the last 2 years, so it's really exciting that it actually came out. We can get this cohort size up to about 1.7 million and do a variety of cross-validation approaches. So if our goal is to live longer, healthier lives by preventing premature death, where do I think the future lies? I see you there, Gil. Genetic discovery for the dual purposes of identifying pharmaceutical targets as well as identifying high-risk individuals. Focusing on coding variation has proven very useful at identifying causal genes right away. Functional fine mapping by encouraging collaboration between genetics discovery and molecular biology, prediction and prevention, and potentially returning risk scores to participants to improve uptake of lifestyle interventions. I think it's still an open question. If you tell an individual you're in the top risk group for heart attack, will they be more likely to lose weight, exercise, perhaps go on statins? Couple of suggestions for how I think we could make UK Biobank even more useful. I think it'd be really helpful to quantify the accuracy of any biobank-specific measurements to standard clinical tests. For example, the UK Biobank released HEAL estimated bone mineral density And we've had reviewer comments saying, well, if you replicate standard bone mineral density approaches using heel estimated bone mineral density, are you sure these are the same phenotypes? So having that correlation with standard clinical measurements would be extremely helpful. I think it's extremely important to have self-reported family history information for more phenotypes, so if survey data is available, I think that would be really wonderful. Adding specialty clinic ascertainment, imputation of more variants from TopMed or other very deep reference panels, and of course, no question, it'll be very important to have full genome or exome sequencing on the cohort. So thank you very much. UK Biobank is a wonderful resource, and I'm grateful to everyone who participated and all the people working on the team to make the data available to scientists. Thank you.